Thank you, Allison, and I really appreciate the opportunity to talk here. I'm honored. I've been very privileged to work at the Simons Foundation for the last decade, and I've known Allison for a really long time, and I've just been super impressed by this foundation. So progress in genetics has been extremely rapid in the last 10 years. Both private foundations and the government have spent well more than $100 million putting together cohorts of individuals and then paying for an enormous amount of sequencing to understand the genetic landscape. We've known for a long time that there's a major genetic component of autism based on things like twin studies. And progress has just absolutely been spectacular. So in the last 10 years, we've gone from really having a very small understanding of the genetics to, for the first time ever, having a list of genes that we have really total confidence that they convey large risk for autism. And in addition to a long and growing list, which is on the right there, we have a list of many more genes that we know are likely to be involved as well. So this understanding of genetics helps us to understand, to answer an incredibly fundamental question, which I think is high on the mind of parents for kids with autism, which for many of them, this helps to answer the question, why does my child have autism? This itself is progress, and I think we really should celebrate it. It's been a very productive 10 years. For many people, this ends a diagnostic odyssey that lasts a really long time, where parents are trying to figure out what caused this autism. But now that we have this list of genes, how are we going to turn it into something that really makes a difference in the lives of individuals with autism? And are there reasons to be hopeful? So what I'd like to convince you in the next 12 minutes, or 10 minutes now, is that genetics is fundamentally changing the way we define and think about autism. Genetics, somewhat paradoxically, may actually help to identify environmental risk factors. And lastly, genetics may pave the way for truly transformative technologies in the next 10 years. So just to make sure we're all on the same page, let's remind ourselves how autism is currently defined and diagnosed. The Diagnostic and Statistical Manual, or DSM, basically defines autism by a list of behavioral criteria. So experienced clinicians see a child and go through a list of questions, essentially, and diagnostic instruments based on the child's behavior. And they involve things like persistent deficits in social communication and social interaction. I think these things are probably well known to people in this audience. Here's how I think genetics is starting to change the way we think about autism diagnostics. And this has really led to a large number of surprises. So on the left, I've shown you, this is from a short review article by Joe Buxbaum. And on the left, I've shown you a list in red of the high confidence autism genes that have been identified in the last decade. And what's really interesting and really intriguing from this list, and you'll see, is not only do they confer a risk for autism spectrum disorder, but many of these same genes confer a risk for intellectual disability, schizophrenia, epilepsy, and including things like congenital heart disease in some cases. So I think this is telling us something very deep and very surprising and potentially very important thinking forward about therapeutics and how we think about the disorder being defined. And what I envision happening, it's already starting to happen, but in the next decade, I think what's going to happen is we're going to augment this definition of autism that we have based on a behavioral diagnosis in the DSM. We're going to integrate that with information that we're learning about the genetics, and the genetics is leading us to the neurobiology, the neuroscience. And we're going to end up at a place where we're going to subdivide what we're calling autism now into functional groups or groups of individuals that are defined not necessarily just by this broad diagnosis, but are defined by shared molecular pathways. And I think an interesting metaphor and an example where this has worked particularly well is in cancer. So in the 70s or so, there was this war on cancer, and people thought of cancer as an entity. And I think what we've been learning over the last 20, 30 years in cancer is that by separating people into different groups with different molecular mechanisms, we've been able to develop therapeutics that work for some groups, but that are of no use to others. And I think this metaphor going forward is going to be very critical for autism. And now, paradoxically, genetics may also help us understand environmental effects. These have been notoriously difficult to study. But we know from the twin studies and from the history of autism that there is an environmental component that goes along with the genetic risk. And importantly, understanding environmental risks might be particularly important. It's going to be hard to change genetics, but if we understood certain things about the environment, we might be able to intervene. So I'm going to just tell you a very brief story. This comes from Mark Zilka's lab at the University of North Carolina. It was a very clever experiment. So what Mark did is he studied, and this is in neuronal cell culture. So Mark studied over 5,000 genes, and he added about 300 different chemicals that are commonly used in agriculture and other places in the environment. And he asked the question, how do these chemicals that are sometimes found in the environment influence the transcription of these genes? And that means, does it turn them on or turn them off? And what he found is that one group of these chemicals actually elicited a change in the genetic pattern, that turning the genes on or off, that looked very much like the pattern that you see in autism brains. And while this is not at all definitive, it's a very interesting piece of a puzzle. So this group of chemicals is called pyrethroids. It gives the environmental studies sort of an additional boost. And I think going forward, this is going to be a really interesting thing. Now, I'm also going to talk about how genetics may pave the way for transformative therapeutic advances, and I'm going to give you a very brief introduction to a pretty spectacular study that was published last year in spinal muscular atrophy. This is a progressive monogenic motor neuron disease. The onset is during infancy, and kids fail to achieve motor milestones, and many of them need ventilators or die at a very early age. This is a very serious disease. You may have heard of gene therapy. This is one of the most spectacular examples where gene therapy was actually very effective, and it involved functionally replacing the gene that has been mutated in this disorder. And it involved a single intravenous, so just into the blood, injection of a virus that carries the DNA to repair this broken gene. And so here's the data. What you see here are 15 kids who got this virus that carries the DNA at a very early age. And historically, the dotted line there at 40, no kids cross that line in terms of developmental outcomes. And they often go down and then die by 18 or 20 months. And what you see here is this spectacular result that all of these kids improved very, very dramatically. And there was a really beautiful accompanying piece in Science magazine talking about this little girl, Evelyn, who received this injection at 4 months, and here she is at 3 years old, sort of running around the house. So I want to make it absolutely clear, I'm not suggesting that we're ready for gene therapy in autism, but I want to sort of take us through what were the ingredients of this success that we might be able to think of as a field. So the first one is that they knew a molecular target, and that's why I think we're focusing on the genetics of autism. This therapeutic advance would not work for muscle weakness. It works for SMA because we know specifically what's going wrong. It involved an enormous amount of work in animal models. So we knew about this gene as a cause about 20 years ago. It's taken 20 years of work to get there. It involved very early identification and treatment. So all of these kids were treated very early at a time when it's likely that the brain is going to be most plastic and amenable to treatment. So I think this is something we need to be thinking of. And it involved having reliable outcome measures. The autism field really needs to develop ways to measure outcomes as reliably as possible. And these are likely going to need to be objective, continuous, and ubiquitous. So really careful monitoring and development, and we're not there yet. Lastly, it's going to need engaged patient alliances. If you're going to need to find people with specific mutations or specific genetic things, you're going to need to have families on your side, and that's where I'm going to just end here with a shameless plug for a major new effort that the Simons Foundation is undertaking called SPARK. Many of the people who are involved in this new project are here. And this involves basically a new way of engaging families, very large numbers of families, in research. It involves, we're aware of all the barriers that prevent people from getting into clinical research programs. So this involves online enrollment. We're also collaborating with a number of clinical sites around the country. It involves genetic characterization just by sending saliva through the mail. And the progress to date has been pretty spectacular. They've recruited over 100,000 people. That includes 38,000 people with autism. And the hope is, just as in pediatric cancer, where the vast majority of kids who have pediatric cancer are involved in study protocols, the hope is that basically everyone with autism moving forward can take advantage of something like this, which has a very low barrier for entry, and be engaged in research. So I want to end with the same quote that I started with. This is from Bill Gates. I'm not sort of terribly fond of quoting Bill Gates, but I think this is a really interesting one. It's basically, we always overestimate the change that will occur in the next 2 years and underestimate the change that will occur in the next 10. I think we're reaching a point in biology, genetics, and neuroscience where there's really some very bold and interesting horizons to go. But the last part of this is, don't let yourself be lulled into inaction. I think it's really critical that every family with autism be thinking in terms of a research setting. There's been tremendous advance in genetics, but there's a lot more work to do. And what we're learning about the heterogeneity and the overall landscape of genetics, we're going to need a lot of people. So I'm going to end there, and thank you very much for your attention.